The overall goal of the following experiment is to perform long-term co-culture of non-typable Haemophilus influenza, or NTHI, with primary human respiratory epithelial tissues using the EpiAirway model. This is achieved by inoculating the apical surface with NTHI to allow the bacteria to adhere to and enter the tissues. As a second step, the inserts are washed every 24 hours with pre-warmed DPBS to mimic normal mucus flow and to remove mucin, waste products, and any cellular debris. Next, the tissues are harvested at the desired time point, and the survival of either total cell-associated or internalized NTHI is determined. Finally, with this method, strain-specific characteristics of NTHI survival within the tissues over time based on viable bacteria counts can be evaluated.
Though this method can provide insight into long-term infections with non-typable Haemophilus influenza, it can also be used in the study of other human pathogens, such as Moraxella catarrhalis. Demonstrating this procedure will be Dr. Dabin, a postdoctoral fellow in my laboratory.
Remove the transport plate from its packaging and prepare to begin the experiment. Spray the transport plate with 70% ethanol and place the plate in the biosafety cabinet. Aliquot 1 milliliter of 4 degrees Celsius EpiAirways maintenance media into each well of a 6-well plate. Then, after removing the tape, open the plate and remove the gauze covering the EpiAirway inserts. Using a twisting motion, use tweezers to release an insert from the agarose. Place the insert into one of the wells of the 6-well plate. If needed, use a sterile cotton swab to carefully remove the agarose from the insert, avoiding touching the membrane. Confirm that no air bubbles are trapped beneath the inserts, and then place the